So hi everyone. Hi, Oliver here. Hello and welcome. Microbehunter here. Welcome again to another Saturday microscopy live stream. Like every time, I'm just trying to figure out if everything is working. Yes, I can see myself as well. There's usually a couple of seconds delay. That's why I always have to check if everything works. And I think sound is okay. Hadrian is telling me thank you very much. I do not want to start the live stream without a sound. This already happened before as well. Yeah, and again, hello all around the world. Today I would like to put a water sample under the microscope. As a matter of fact, you see over here under my stereo microscope, you already see a plant with something growing on it. I would like to have a closer look at that today. Yeah, it's Stentor and Vorticella, among other things that I'd like to explore together with you today. Yeah, I see that a lot of people have already joined in. Usually the first couple of minutes, I will do the following. I will just say hello all around the world. This gives people time to join in, yeah, so they don't miss a lot. Yeah, I'm just going to go through the chat very quickly. Hello from Athens, Greece, from Oregon. Yeah, and... yeah, and from northern France, from Germany, from Finland, from the United States, Slovakia, UK, Essex, UK, from Germany again. Hello from Linz. Hello. Yes, there are also some people joining in who I know from school as well. Okay, from South Ireland. Okay, sound is good. Netherlands, UK, Italy. I love it. If there's so many people all around the world. New York City, Netherlands, Illinois, USA. Okay, Georgia in the United States. Hello from Wales. Yeah, also quite close by from where I am. Barcelona, and also from England. Jesse has joined. Thank you very much. I'm going to also show a couple of videos that Jesse sent me over link today. So just a very short Oregon again, short overview of what I plan to do today. I'm going to put a water sample under the microscope like the one that I've got here, and I would like to then later on, if there's a little bit of time, also show you quickly a little bit how my mold is doing, because last week I put some Penicilium fungus under the microscope, and now I can see over here that the petri dishes are all overgrown with fungi. So I'm going to show you how this looks like. Then basically we're going to take it from there, and if you have any questions or so, please feel free to type them into the chat, and after approximately 1 hour I'm going to try to again answer the questions. Okay, yeah, so from Maine, USA, from the Philippines. Okay, Montreal. Okay, great. So background information. Today in the afternoon, it was spring is slowly, I'm in Central Europe, so slowly it's starting to become a little bit warm. And I would like to show you where I've been. I was actually visiting a local pond. Yeah, this is basically, that's the place that I've been. It was reasonably warm, so I spent around half an hour, 45 minutes walking around the pond. Essentially, lots of ducks were there as well. And I wanted to collect a water sample. And of course, water samples, when I say water samples, I'm not talking about water samples. I'm actually talking about algae or something solid. And I found those plants here. And those plants are always a good specimen because not the plants themselves are so interesting, but rather that's the stuff that is actually growing on it. So I collected a couple of those plants, and that's basically what we've seen, okay? Because many interesting microorganisms, they like to grow on a surface, and decomposing wood is a possibility, rocks, of course, but for whatever reason, maybe it's the oxygen production or simply the fact that their plants are primary producers and also produce a lot of organic material. There are plenty of microorganisms usually growing in a biofilm on the surface of a plant. Okay, yeah. So this is basically the thing that I took along, and that's basically what you're seeing right now here under the microscope. Where is it? Here we go again. Okay, so that's basically this little jar that you see here on top. That is the thing, and for whatever reason, I'm not seeing that's a little bit sad. All of a sudden, my the camera died on me. That's always a little bit of an unfortunate thing. Why is that? Okay, this is a little, here we go again. Oh, lucky, lucky, lucky. Okay, so, and I'm going to quickly show you of what we're actually seeing here. So that's on top, that's the plant that I picked just a couple of hours ago from the pond. And under the stereo microscope, we can actually zoom out a little bit so that you actually see this. You can actually see how this looks like. Yeah, and I'm not so much interested on the plant, but rather on the stuff here, the area that's growing on here, right? You can see this fuzzy layer here. These are vorticella, or at least they appear to be vorticella. And those little things that are growing over here, I have to zoom in. These are stentors, and there are ciliates, they're protozoans, and they're a single cell. And yes, when I say a single cell, this should be surprising because actually they're pretty huge for individual cells, right? So these are stentors here. They're also known as, in German, Trompetentierchen. These are not animals, even though it's or also trumpet animalcules. Yeah, so because they have a shape of a trumpet, right? And what I want to do is I actually want to pick them off and I want to put them under my compound microscope. And you can see also in the background, there's lots of other stuff also floating around here. Okay, let's rezoom here. Yeah, not rezoom, but refocus. There's just some stuff floating on the surface. Essentially, these are quite nice to look at. What I have to do is I have to use a pipette and I have to pick them off and transfer them to a microscope slide, right? And then hopefully we're able to see them a little bit better. Of course, I tried that already and it works surprisingly well. It does take a little bit of patience, however. And you see that there are also some other microorganisms floating around in the back. Okay, so every now and then, for those of you who are new to the system, every now and then I'm going to pause this live stream. I mean, you continue to see me, but I'm going to make a short break to simply read the comments that you've got over here. And then, I'm going to answer them. If they're a little bit off topic, the questions, then I'm going to answer them later on at the end of the live stream, okay? But if the questions are directly related somehow to what I'm talking here, then I will basically try to attempt to answer them right away, okay? Yeah, please include, as like some of you are doing, at Microbehunter or something like that, so that it's easy for me to see, yeah? Yes, yes, Alexandre is basically also saying that he observed also a stentor or vorticella. I remember we had this conversation, I think, last week or 2 weeks ago. So what I'm going to do now is the following, is I'm going to now move over to my stereo microscope, which is just right next to me, but you're not able to see me then. And I'm going to try to use a pipette. Where's my pipette? Here is a pipette, and I'm going to try to pick off. I'm going to try to use this pipette and I'm going to try to suck off the stentor and using the pipette, and then hopefully I'm able to transfer it on a microscope slide. So what I would like to do, however, before I do that, I'm going to I want to first, because I do not want to damage the stentor. So I just want to show you here, where is the desk view, okay? So I've got a pipette over here with a disposable tip, okay? And I would like to make the opening, the hole, a little bit larger by cutting off the tip here, okay? Just in case that the stentor or whatever it is, that will not become damaged as it's being sucked up, make it a little bit bigger yet, as it's sucked up into the tip, right? If it's too narrow, then it might become damaged. But I think it's going to be fine. Yeah. So that's a couple of questions here. Are stentors regenerative? Well, I mean, they're single cells, I think if they're damaged to the extent that the cell's content flows out, then they don't regenerate, right? Normally, simple animals like certain worms or so, they are quite regenerative, because if you split them in half, then they're going to simply grow again. But if you split single cells like stentors and other microorganisms in half, then essentially they die, because the cell flows out and they lose their cellular integrity. Okay, so I hope again that I'm able, yes, here we go again. So I'm going to now do the following. I'm going to switch off my face, okay, so that you get the full picture. So yeah, I'm still around. You should still be able to hear me, but I'm going to switch off now my face. So here we go. I hope that you're still able to hear me. And I'm going to move over the microphone as well. And I'm now going to try to, yeah, pick off some of those, yeah, stentors under the microscope. So, and I'm now looking through here, and it's a little bit difficult for me. I need more light. Okay, and oh, I think I lost them. No, I didn't. Where are they? Okay. The problem is the contrast isn't the very best. Okay, there are lots of vorticella here. I think where was this again? Okay, I've got a little bit of a problem finding them. I think, here they are. I have to turn this around because I'm right-handed and I need to, yeah, so it's the bottom side of the plant. Don't see it here. Here we go. Okay, so, and now let's give it a try. Where is the tip? And here it is, and I'm going to try it with a little bit of luck. I don't know if this worked or not. Okay, but I now picked up. I went very close to the leaf, and I picked up something, and I'm now going to switch over again to the desk view. Here we go. Let's remove the cable from the microphone. Okay, and let's see. This is I tried it already before, and it worked quite well, but I don't see anything here. So I might try it again. Okay, I might try it again. This is a little bit a question of patience. Okay, let me try it again. Here we go. Just a second. I'm just carefully moving the. Yeah. No. The stentors are still. It's still here. Just a second. Where is it? Okay. I don't know if you've. Yeah. I don't know if I've got it. I don't know. I'm going to quickly. This is a little bit of a very needs a lot of patience. Let me try it again. It was kind of surprising because just before it was quite easy, and so. Maybe now I'm a little bit more lucky. So let me show you again the desk view. Yeah, I collected now 3 water droplets here, and I'm just going to put it now under the compound microscope, and let's just try our luck. Okay. Let me see. Let's try it with the low power objective. Ooh. Yes, that's actually already that's one. That's one already. Just a second. Let's remove this here. Yes, as a matter, it doesn't look like a trumpet. That's it. Okay, that's one of them. If it doesn't have the correct shape, this is because it retracted. Yeah, but that's one of the stentors. Let's see if I can actually see another one over here. So the first one was actually that's the second water droplet. It's the second water droplet. I don't see anything here. And the third water droplet, let's see if the third one, also nothing. There's lots of goo over there on the side. But actually, I'm kind of happy that on the first one I was actually able to find something. Kind of surprising. I thought on the first one I didn't catch anything. Where is it again? Maybe it's somewhere in the shadow now. Okay, so what I might want to do is I might want to add a cover glass because it's very dark now because of the refract. Ah, here it is. You actually see it here now, right in the center. Here it is. It looks almost like dark field. Okay, so let's go up. No cover glass now. Okay, for this reason it's yeah, and I do hope that it's we're able to see it extend. So what I'm going to do now is I'm going to add a little bit more water so that it doesn't become crushed, and I'm going to add a cover glass now. Okay, so yeah. Usually what happens is that after a couple of minutes, if we're lucky, it will settle down and it will start to extend into its typical shape. It's very blurry right now because there is no cover glass on here. Okay, so let's try that. Okay, so where is the, oh gee, I have to find always the desk view again here. So let's remove the microphone cable. Okay, so here we go. Okay, so it was the first one here. So a bit more water. And then maybe we can also try some Vorticella later. Let's give it a try. It's only one cover glass, I hope. And and we're going to put it under the microscope again, and now I have to find it again. Because usually every time when you add a cover glass, of course. So we are now going to go down to the low power, and we have to reduce the light a little bit. And now let's try to find it. Okay, so let's usually what I do is I try to scan, yeah, the sides here, and yeah, here the next one. I don't know what that is. Then the next one, I usually very systematically you try to. You see, there's a little bit of movement already going on. There are other microorganisms, of course, also around. Here it is, here it is, here it is, here it is. Okay, so once we've got that, we go up with the magnification and we refocus. Okay, yeah, and yeah, it's slowly starting to become long again. Yeah. So, and you know what? Why not? 20 times. Here we go. Yeah, that's actually quite nice. Yeah. So what you actually, yeah, are seeing right now that it's now moving around, obviously, right? And occasionally when we're lucky, it actually will attach, and then I've seen it that becomes very, very long. Yeah. Maybe, yeah, it depends. Maybe it doesn't want to connect or attach itself because, yeah, there is not a lot of space now beneath the cover glass. Yeah. So, yeah. So Alexandra is now confirming it's not what I saw 2 weeks ago. Yeah, yeah. And in Stentors, what you have is there's this typical ring of cilia here at the mouth. Yeah, and then basically when it attaches, this stalk, it can become very long. But similar to the Vorticella, it's also able to retract. Yeah. Right now, it's really limited in its movement. But yeah, I don't know. I'm going to add a bit more water. A bit more water here on the side of the cover glass, maybe to make the cover glass go up a little bit, so to give it a little bit more space. Yeah. 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 Did you just see this here? This seems to be a contractile vacuole. Yeah. This was yeah. Usually ciliates they have those. Basically, they will pump out water. Yeah. So, but let's try to follow it a little bit. Yeah. Yeah. So I'm going to add a bit more water to the side of the cover glass to ensure that not only that it doesn't dry out. Yeah. So the question is, oops, I bumped now into here we go again. What's the size? Okay, you know, that's actually a good question. I'm going to show you something. We're now here at the edge of the cover glass. Yeah. So let's see. And let me see if I have got it. I've got this. Just a second. Give me a second. Give me a second. Okay. Just want to show you something here. I'm going to now add a so-called a micrometer slide. Yeah, so I'm just going to show you here. Look at the top again here, okay? At the stage. Look what I'm doing here. Okay. Okay. So that's kind of dark. Why is it so dark? That's so I'm going to now add the micrometer slide. Okay. And let's have now a look at the micrometer slide. Now I have to find, the problem is I have to find it. Here is something, here is something, here is something, here is something. Okay, let's see. Okay, let me quickly have a look. So, okay. So it's upside down. One division is 0.1 millimeter, okay? So one division is a tenth of a millimeter, which is 100 micrometers, okay? So one division, so one line is 0.1 millimeter, okay? So let's have a look. Where are the divisions? Yeah. So essentially, this here, from here to here, is 0.1 millimeter. Okay? Just a second. Okay? 0.1 millimeter is one division. Okay? So let's have a look again. So this means that let's copy this. Okay, so I've got now two arrows. I'm going to place two arrows next to each other. That would be a tenth of a millimeter. Yeah, okay. And let's see if we're able to find the vorticella again. I have to, here it is. Ooh, I don't know. It's not moving anymore. Okay. So this basically means, yeah, it's a tenth of a millimeter. Yeah, so when it's extended, maybe it's 10 times the distance. So yeah, it can be actually, yeah, I don't know, fifth, maybe even up to a millimeter long. Okay, if those two arrows next to each other are tenths, and it becomes, let's say, 10 times as long, then it can actually become, yeah, a full millimeter long when it's, yeah. Basically, or maybe even longer when it's fully extended. Yeah, so that's a little bit possibility of when using a micrometer slide. Now I don't know why it's not doing so fine anymore. It's a little bit of a it doesn't look healthy anymore. Maybe it's slowly getting crushed by the evaporating water. I don't know. So I'm going to add a little bit more water here. Okay, I don't know where it is now. Yeah, I'd have to find it again. Yeah, here it is. Oh, okay. Swimming away again. I did make a video, which I'm going to show you later, of one that's extended, how it looks like. Yeah. But maybe I'm just going to try to pick off another one from the from the water sample. Yeah. So yeah, so that's kind of. I'm a little bit. I don't know why it's so dark here at the top now. I don't know. Let's see. Somehow the camera seems to readjust this a little bit. It's kind of strange. Yeah. Okay. Yeah. So see, the camera seems to readjust the whole thing. So let's have a look here again. So, but you know what? I'm just going to show you briefly one of the videos that I made. I don't know which one it is. Yeah, that's the one that I made just today in the afternoon. Yeah, this was one of those Stentors after it was extended. It looked quite round before, but then it settled down and it extended. And look, this is quite remarkable. Yeah, it's extremely long. Yeah. Yeah, so you can also see the cilia here. Yeah, front beating and yeah, moving the water around so that it's able to eat and to feed. So I'm just changing now. I did not edit the video a lot, so I'm just changing right now the magnification and everything. Yeah. But you can see now on the outside as well. Here, yeah, that there are these tiny little cilia, those tiny little hair, yeah, as well, and pretty big ones over here. Yeah, and somewhere in here there must be the mouth where the food is being sucked in. So I think quite a nice little specimen here. Okay. That's an interesting one here. A poor man's micrometer slide uses 0.5 millimeter HB lead at 4 times, or just a ruler. Yes, this is actually also something that I've done. Also with my students in school, you can actually use a regular ruler as well. And then you have to do a little bit of math, right? You measure out basically the distance of 1 millimeter on screen, and then you can do some direct proportion calculations, and you can essentially also calculate, yeah, then the size of different objects. Yeah? So is a Stentor a bit like a Euglena? Yeah, or does it use algae sometimes? Well, I mean, it's a protozoan, it's a ciliate, right? Euglena is a flagellate. I mean, it's a different category again. But these are single-celled eukaryotes. In that sense, they're similar. Euglena is also able to do photosynthesis. I don't think that Stentor is able to do that. Ultimately, even though they're called little animals, they're not animals. They're a category on their own. They're protozoans or protists. Yeah? So, yeah. Yeah, here again, we are able to see those tiny little cilia much better. But what I'm going to do now, so that's basically just for your info, that's a video that I just made before. Yeah, so I can actually, yeah, that's the foot. Yeah. Yeah, that's something I wanted to show you here. Look what happens now. I think I was able to catch, boom, and it's gone. No, it's not gone. It simply retracted so quickly, yeah, because evidently something disturbed it, and then it retracted so quickly, and it got, more roundish again. Yeah, so it's kind of fun. Yeah. Yeah, this Alexandra is also commenting over here to Marcel. I use indeed OBS Studio, which is a free program. Many people who are live streaming use that. I highly recommend it. I use OBS Studio also for simply recording videos. Because I've got my cameras all connected, I've got my different scenes, and I can switch the cameras back and forth. It's really convenient, and I can combine different cameras together. So I'm actually also using it for making regular recordings. So let's go back a little bit to the microscope view here. I'm going to not delete, but I'm going to remove again. Here it is again. And let's see. Is it here? That's the live view again. Let's see if it's slowly starting to extend again. And then I'd like to try to pick off a couple of vorticella and see if this actually also works. Yeah. Okay. Yeah. So. Yeah, in case, for those of you who are new or who don't know the background yet, very often is a question what people want to know is what gives the background, why is the background so blue? I can change the color around like this as well. That is because I'm using a technique called DIC, which is called differential interference contrast. It's quite a mouthful. So when most people don't have that, and most people, if you just observe it, you will see it like this in regular bright field. So these are actually the natural colors. Yeah, these are the natural colors. And okay, I'm going to quickly remove the second arrow as well. Yeah, and see now it's slowly starting to extend. Yeah, maybe it takes time for it to feel a little more comfortable. So these are again the natural colors. Yeah. Okay, if no micrometer slide, can you remind us about the stage scales? So it is also possible, indeed, maybe I'm going to make another separate video of this, but I have to reset the camera. Many microscopes have a so-called a micrometer scale or a scale on the stage, and with that it's also possible to estimate the sizes. Yeah, so yeah. what I consider fascinating is look, look, you see the stentor is one cell, but then you see also other cells floating around, yeah, on to the left of it. These are also individual cells. It kind of shows how cells can be of a vastly different size. Now it's starting to slowly extend. Beautiful. Okay. Would it be advantageous to use slides with spacer rings of 0.3 millimeter thickness to give Stentor a bit more freedom of movement? Well, I did not use spacer rings, but I tried today in the afternoon to use more water. And then I had the problem that the Stentor kind of also moved up and down, and I had a problem with focus. Everything's got some advantages and disadvantages. Or, for example, if you just leave it in a droplet of water without a cover glass to give it more freedom, then basically, yeah, you have the problem that sometimes it moves out of focus. So that's a little bit simply the trade-off, so to say. Huh? Yeah. So now it's almost fully extended. Yeah. Okay, now let's use this opportunity and let's do the following. I'm going to quickly show you now phase contrast at 40 times, simply for the fun of it. Okay. Need way more light. So that here is now phase contrast at 40 times. Yeah. It's also quite nice. Phase contrast microscopy allows you to view transparent objects, or let's put it in other words, transparent objects in a greater contrast. So in other words, phase contrast microscopy is able to convert differences in refractive index into brightness differences. Yeah? So these are, of course, not the natural colors anymore. Generally, phase contrast does not always look as beautiful, maybe, but sometimes you're able to see more details that you otherwise would not be able to see. Yeah? So, yeah. Occasionally, I'm just reading also the comments. So if I'm not talking, yeah, then yeah. So now, quite nice. Now it's again fully extended. I've observed this already several times. When I pick it off from the plant, it's irritated. It moves around quickly, but then after a couple of minutes, usually it starts to settle down and it starts to extend. Yeah? So let's move back again to the regular. So, and I have to close the condenser again, otherwise it's way too bright. It seemed to have retracted again. Yeah. So I'm going to add yet again a little bit more water. Sometimes the microscope, the cover glass then starts to float. Okay. It's kind of remarkable how it's able to change its shape. Huh? So I'm just checking the time. Okay, I've been talking now for half an hour. I mean, maybe I'm going to give it a couple of more minutes. And I do want to look at some of the Vorticella, but I don't know if I'm able to pick them off the same way. Yeah, and yeah. So occasionally I'm reading again your comments. Okay. Yeah, and it's swimming around again. So, okay, you know what? I'm going to give it a, you prefer dark field. You know what? I can also do improvised dark field, but only using the 4 times, my 4 times objective, okay? And this is possible because I'm simply going to use the phase ring, as a dark field filter, not intended, but this would now be dark field. Yeah, so I can only have dark field. Unfortunately, with my 4 times objective. Yeah, so I'm right now using the phase annulus for phase contrast. Can also be used as a dark field. As a dark field filter. Yeah. Okay, and at a higher magnification, you see I'm losing the dark field effect. And let's need to close this again here. Let's go back to this one here. Okay, I guess you get the point. Okay, and I would like to now observe, hopefully, be able to observe some other things that I've found in this plant. Okay, so I'm going to switch back again to the stereo view. Where is it? Here we go again. Okay, again you won't see my face anymore. Let me just, yeah, now you should. Yeah, okay, but I'm going to put it away, and I'm going to now try to pick some Vorticella, and so somebody, yeah, someday I hope to win the lottery to get DIC. What you can try to do is Reinberg illumination and oblique illumination. It's yeah, so that's sometimes also it's a little bit gives similar effects. So I'm just going to see, I have to center this a little bit so that you are able to see this as well. So now again, this is a question of ooh, I cannot reach it quite easily. I don't know if I don't know if I was able to catch anything here. But as so often, everything is a question of trial and error. So, and where is the scope view? And what I usually do is here it is again. Look, look, look. I'm not going to move it away. It's slowly extending again. Yeah, it's slowly extending again. Yeah, before I put in the other microscope slide. So I'm going to put in the other slide now. You can see this at the very top here. I'm adding the others, and without a cover glass, and hoping that maybe or not nothing. The Vorticelli, they seem to be fairly solidly attached, because I don't see any. I don't see any. There are some other microorganisms. Look. Don't know what that is, but no, that's not what I wanted. What's this? I don't know. Okay. This is dirt. Okay, so not so lucky. So what I'm going to try is I'm going to try now something slightly aggressive. Okay, just yeah, basically simply for the sake of trying it out. I'm going to try now something slightly aggressive, which yeah, I'm just going to do for the sake of experimentation. So let's put this away. I am going to pick. I need some tools. Let's try this. And I'm simply going to scratch the surface and try to collect some material this way. Actually, I hope to find Hydra, because in this lake a couple of months ago I did pull out a water plant where quite a few Hydra were on it. No Hydra this time, but some Stentors, and I don't know if this is actually now going to work or not, but this is a little bit this experiment that I wanted to try to see if this kind of yeah, has done the trick. The scope view. Okay, I need to flip this away again. And let's see if there is there's more stuff here, but certainly not as. Ooh, a nematode worm. Okay. What was this? What? There was something bigger swimming around this. I'm going to remove the filter. So this is now regular bright field. I'd have to zoom in a little more. Use a nematode worm. This was so... yeah, here. I did collect a, a Stentor. Okay. So this is now without cover glass, so plenty of freedom of movement here. And what again? Worm. Are the leaves too thick to put under the microscope? Yes, they are. But I can show you how this looks like because they're also not flat enough. That's the problem. Yeah. So if I want to go up with the magnification now, I mean, that's a little bit disappointing that I was not able to get any Vorticella off the leaf. But maybe we can have a look at some of the other microorganisms that are there. And for that, I do need a cover glass. Okay, but of course, every time, you know what? I'm thinking. Let's try the following. Okay, let's try, and this is probably not going to work well, but I'm going to try it in any case. I'm just going to indeed take one of the leaves here. I need to grab myself again. I'm going to indeed take one of the leaves here. Up, I dropped it. And I'm just going to indeed cut off a piece and put it under the microscope. However, this is probably not going to work well. But maybe, because it's very thick, but maybe we're still able to see something on the side. So let's try it. Okay, I need to dry wipe it. Let me quickly change to this microscope view again. So let's give it a try. So this is now the leaf, right? Okay, so that means it's way too dark. As you can see, I'm going to really turn up the brightness. Okay, it's a little too bright. Okay, so that's now the leaf. Yeah, let's have to move up here. So to get a better view, and you see the leaf itself is too dark. Let's try like this. Yeah, of course, on the surface itself, we're not able to see anything. Not... this light got stuck. Trying to get this adjusted is very difficult now. Yeah, not a satisfactory solution here. Nah. No, you see, this is, I think, yeah. This is a Vorticella, what you see going around in a circle. Okay, so this actually is a Vorticella. Very lonely one with its stalk. Okay, yeah. Those Vorticella are actually very fast organisms. They can contract quite rapidly. And this one over here is, yeah, detached. Oh, what is this? Let me have a look here. Water crustacean. Where is it now? Here it is. Okay. Okay, that's also nice. So you see, you see this. If you want to observe water samples, you have to, you have to look for specimens growing on a surface. Yeah, the biofilm is usually the best place. You see the slide. If you look carefully, the slide doesn't move because there's water beneath the slide and that's why it got stuck. Yeah. So yeah, I'm going to do the following now. I'm just going to go to the desk view again. A question here. I thought that stentors were sessile, but here this one is able to move around. Do stentors change location when food is scarce? Well, I don't know. Good question. Why not? I would say, but generally, what they do is they generate a water movement, a water stream, and this way they're able to collect bacteria and other debris and small particles as a food source. So I don't know if they're actually moving around when food becomes scarce. I guess it's possible. Yeah, but. The question then I would say is if food is scarce, then maybe they wouldn't have reproduced in the first place. Yeah, and those microorganisms, they don't need a lot of food anyway. So yeah, so let's see how we're gonna do this. I would actually like to see a little bit this under the stereo microscope again. Let me quickly check, and then I would like to show you some of the fungi from last time. Here we go. Again. Let me refocus. Okay, and so that's a little bit the thing here. Yeah, that's probably better. Yeah, so this leaf apparently did not have a lot of things growing on it. Okay, Jesse researched something. Google suggests that they can move to new locations if needed and will free swim when necessary. Yeah, so this leaf over here is not the best one, I think. I'm going to give it still, nevertheless, another try because I do want to see those colonies of Vorticella. Should look quite nice to look at. But yeah, maybe I'm just going to try it here again. I guess it's also maybe a bit a question of luck whether the leaf actually now contains sufficiently high number of microorganisms or not. Let's check again using the stereo microscope. Where is it? Or was it this one over here? I don't know. I mean, you see there's lots of movement going on in any case. At least I can see it, but yeah. I'm going to take the whole beaker again. And let's refocus. You see, there are. Yeah, here. I use the arrow now to point. You see over here? Yeah, I'd like to get this somehow. I'd like to get this off, but that's a little difficult. Let me try again. And then what I will do is, what time is it? Yeah, and then I'll quickly show you some of the fungi from last week. So where is it? I cannot reach it, unfortunately. Let's try here the. Where? Let's hear it right at this stem here. I don't know if this actually did anything. So let's quickly check. I have my doubts. But I mean, you get the idea. That's a little bit part of the, how do you say, the fun of the whole thing. Simply to, yeah, that's a Vorticella, finally. Finally, I've got one. Let's do this here, this here, this here. Turn on the light. Whoop, and it's off again. Don't run away. And it was out of focus. Here it is. Okay. Okay. You can, yeah, very nice. So you can actually see how it's able to contract its stalk. Yeah. Yeah. So, while the stentors are called trumpet animalcules, those vorticella are referred to as belled animalcules because they have a bell shape, and they're also protozoans made of a single cell. Yeah? And actually one of the fastest ones, because they're able to, the stalk that they have, they're able to contract it very quickly. Okay. Yeah. It's apparently one of the fastest movements known in biology. Yeah, I made videos, on this, and you can actually see how the particles are actually, streaming towards it. It's able to generate a vortex, and that's why it's referred to as a vorticella. Okay. What is? I have an idea. I'm just reading as well. Stentors have a remarkable regenerative powers. A small fragment, less than 1/100th of the volume of an adult, can grow into a complete organism. That's interesting, really? So this basically, you're able to take a fragment of a stentor, even though it's less than a cell. That's interesting. Didn't know that. Because normally regeneration is of course possible with multicellular animals. Because a few cells are then sufficient, and then they will divide, and then you basically get a new animal. But that is also possible with single-celled. Because essentially, if you take a small fragment, and this fragment does not contain DNA, then I wonder how it's able to regenerate. But then again, nature is extremely diverse, and one should not be surprised. So this is just for your info without cover glass. So I'm not able to go higher. So I'm using my 20 times objective right now. If I were to go higher, then of course my objective might become wet. I don't like that, obviously. So I'm just going to leave it at this magnification. Yeah. So, and having said that, now you could actually see how it contracted. Yeah. Now it attached itself to surface. Yes. Okay, maybe we're able to see it again, how it contracts. Yeah. Fascinating. Huh. So occasionally I'm reading again the comments here. Okay. I think the stentor fragment needs to contain part of the macronucleus. Yeah, the protists, they have those macro and micronuclei. So maybe if some of the DNA is present, then it's able to survive. What does the cella part of the word cella mean? Arcella. Okay, here now nicely extended. You need to save for micromanipulator, only $20,000 or so. Yes. As a matter of fact, you're going to laugh. I actually considered making my own micromanipulator, using 3D printing one maybe, and maybe by making some, I don't know, by pulling strings maybe, and bending, the little, how do you call this? A little piece of plastic. I gave up on that. Micromanipulators are indeed. However, what I have done already before is, and this is possible if you really are very patient, is to use indeed a microliter pipette and then to pick off individual cells. That is possible. yeah, of course, you're going to pick up a lot of other things as well this way. But at least I was able to isolate this way already some, yeah, water bears, for example. And yeah, when I collected so-called the face mites, then I also was able to isolate them using, yeah, a microliter pipette. Yeah, and it's going around in a circle. Making me dizzy already. Okay, you get the idea, right? Let's go down a little bit with the magnification here. Where is it? Here it is. Okay, it's too bright. So at least we've got that one as well. Okay. So next comment. So then what I'm going to do now is the following. What time is it? Okay, yeah, almost 1 hour. Because I'm going to soon, in a couple of minutes, start to answer some of the questions and do some other organizational things. I want to switch topics a little bit and just want to show you, yeah, the plates of the spores that I started last week. Okay, so what I'm going to do first is I'm going to quickly, I'm going to quickly show you. Yeah, here the questions are already coming here. Thank you very much. There are lots of comments again, over almost 2 pages of comments. Thank you. So I'll quickly in a few minutes go through those. I want to just show you very quickly because somebody wanted to know how my slime molds are doing. Yeah, not so great. I threw out the others and I kept one of them here. And basically, yeah, it's very slimy, but also because there is quite a bit of bacterial contamination here as well. But maybe I'm able to save this one over here. Yeah, so this is basically the slime mold. And what I've done is I've got several others on filter paper, which I dried for long-term preservation, right? But this one over here just kept going a little bit. So it's not a lot, honestly, not a very, how do you say, a good one. But I've got others where the whole plate was full with a slime mold because I really amplified the growth. And I dried it so that I have the specimen saved for later, okay? But what I wanted to show you is the following. This is going to be scary. Careful, I'm going to show you something very disgusting now. Okay, and look, these are look at this. These are now the plates that we've prepared last week. Okay, basically with the fungus from the orange. This is the place where I pressed the orange directly on the filter plate paper. And if you were not around last week, let me quickly have a look. Yeah, I just want to show you. This is what I have done last week. Okay, I've collected some of the mold spores here and I transferred it on some filter paper with a little bit of orange juice. And then basically I waited. And that's the result that we have now, right? So that's all I've done. Yeah, so this was last week's. Yeah, so a little bit of orange juice on filter paper in a Petri dish. And this here is the result. So now you want to know why. What's the point? Because I'm going to show you, and unfortunately it's not ready yet. But I wanted to actually observe the spore germination. So I'm going to show you as well what else I've done here. Here inside this box here is I put some slide. This is wet tissue paper here on the bottom. And here is a slide, and I put some of the spores with orange juice on here. Why did I put it on wet filter paper? So it does not dry out. Okay, so I wanted to simply keep it, yeah. And let's wipe the moisture away. And the idea is, or was, to actually observe how the spores germinate. Here we go. I was already scared that it's not working. So, and let me see if I'm able to find it. Unfortunately, it's not ready yet. And which surprised me a bit because I thought that they were going to germinate quite quickly. I'm just going to look for the place where there are more spores. Okay, so you see them over here, those little dots, right? These are the spores mixed with orange juice. And this was maybe 8-10 hours ago, and they are not growing, which surprises me, but maybe it's not so surprising. because for two reasons they might not grow. First of all, might be a lack of oxygen. So you see the spores are still here, right? Might be a lack of oxygen beneath the cover glass. And number two, they're completely surrounded by orange juice. And on the filter paper, actually the spores are surrounded. They're on a solid surface. Maybe that's the reason. I don't know. Okay. Yeah, eine feuchte Kammer, yes, indeed. Yeah. So what I've done here is I simply put some wet tissue paper in there. I've also 3D printed a little rack so that I'm able to replace the slides on this rack so there's water on the bottom. It's also possible. Yeah? So this is basically just shows that the spores are not yet germinating, and I wonder why. And because now it's springtime, at least here in Central Europe where I am, also want to observe the germination of pollen. That's also something that I've was semi-successful in. But I would like to make this a little bit more reproducible. But in any case, those fungi are not yet growing. And that's a little bit a pity. Yeah. So I'm reading again your comments. Okay, yeah. So that's simply something I just wanted to keep you a little bit updated. That this is something that I'm kind of working on. Yeah? So could the acidity of the orange juice be an inhibitor? I do not think so. Paul is asking, and the reason why I do not think so is because over here I added orange juice to the filter paper. Yeah, and it actually worked quite well. So it is not the acidity itself, because otherwise this one also would not have worked, because I simply added orange juice here, and then But I indeed think that maybe it is the lack of maybe a solid surface. This could be, I don't know, or maybe the direct exposure to the air. I don't know, or maybe it is too moist because it's surrounded by liquid. I don't know. So essentially, that's something that I still have to figure out. Yeah. Did we ever find out if the chlorella algae tablets came to life when left in water? No, not yet. Okay, that's something that we can also try. Okay, yeah. Thank you for reminding me. Okay. So what I'm going to do now is I still, from last time, I promised you to show you something, okay? And also Jesse also sent me a couple of links. So what I'm going to do now is, just a second, I need to, very quickly, maybe Jesse, you can also comment on this. What I'm going to do now is, where is the Windows here? Yeah, okay, this is from Reddit, okay, and kind of a couple of very interesting things here. So this is now a screen capture, and it's an interesting blob here. And so there are a couple of interesting things, and later there's also a video over here which shows how an amoeba seems to eat and engulf something. It's quite an interesting one. Yeah. So I would like to, so this is something in Reddit, R microscopy, that is the subreddit. Yeah. These are, he's Jesse, thank you, sure. These are Amphizonella amoebas. Okay. The first one we named Blobby. This was before we identified them. Yeah, that's quite interesting. Jesse, maybe can you explain a little bit? I suppose you used dark field microscopy here. Yeah. So it's kind of interesting. As well. So these videos are a little bit longer as well. So I'm going to move forward because I like the one where you could actually see how it eats up. Over here it eats up a whole bundle of algae. So you see the amoeba arriving from the lower. Oh, wow, a donation. Oh, thank you very much. First contribution towards a micro manipulator. Thank you very much. Okay. Yeah, so you see that the amoeba is not engulfing, yeah, what seems to be some kind of cocci or algae or whatever. Yeah, it's quite cool to observe. For your information, in case you're interested, I like to show those videos also yeah, to my students in school, especially when we talk about endocytosis, for example, and yeah, engulfing yeah, of bacteria by, for example, by phagocytes of the white blood cells in our immune system. Yeah, sometimes I use amoeba as an example here on how they actually engulf, how this actually works. Yeah. Yeah, okay. Oliver, you will want to go back to the first video and see the second half when it evacuates its food vacuoles. Okay, I will do that. Just a second. I'll quickly go back to the first video. This one over here. Let's wait a little bit. Let's be a little more patient here. So, and then something seems to be happening. So these are incredibly slow amoebas. So these videos are 1 hour condensed into 2 minutes. So this is a massive time lapse video. Okay. Here it is. Look how it's dumping, how it's dumping the contents of the food vacuole. Yeah. So what they do is what they often do is by the way, Penicillium, not an amoeba, but it also does this. It takes in food and then it digests it, and the thing that's not needed is basically dumped out again, pooped out, so to say. Yeah. So and there's another video. Again, if you want to watch everything with comments and so on, then this is yeah, you can actually go into Reddit. So these are just this is so cool. Yeah. Look, the pseudopodia. These are the, yeah. It almost looks a little bit three-dimensional even. Yeah. The third video is 31 hours in 6 minutes. Unbelievable. Approximately 400 times speed. Don't need to watch the full video, but it has a bunch of yeah, feeding as well as failed feeding attempts. Okay, that's interesting. Can you please tell me how did you actually do that 31 hours without the microscope slide actually drying out? This would be how could you actually stabilize everything so much? And I suppose you took individual pictures and not a video as such, or how did you do that? Yeah, this is yeah. Can you believe it? 31 hours. Okay. Yeah, so this is basically, thank you very much for this. If you're interested in this, please go to Reddit. The videos are publicly visible. Thank you again very much to Jesse for pointing this out and for making those videos. I also want to, before I go to the questions, I want to show you another video. Okay. Yeah, not on a slide. I keep them in a shallow dish with 2 milliliters of water. Okay, it's a video at full HD, 30 frames per second. Wow. Okay. Interesting. Okay. I then take it all and condense it down into DaVinci Resolve. I also use this editing program. 100 gigabytes of video footage. Wow. Rodney the Rotifer. Okay, thank you very much. I'm going to move on, otherwise it's going to be a little too long here. Because I did promise you as well, yeah, because, not this one here, but this one here, Frog Spawn Day 1. This is another video that was sent to me by Dave Lewis. And already 2 weeks ago, we had a look at the development of a tadpole. And this one here is now the first stages of cell division. And I think this is also a very nice video. You see basically a two-stage frog embryo and see how the cells are dividing. Yeah. And yeah, so what we're seeing here is the first stages of frog development. And you can see that now the cells are starting to overall, it's not becoming bigger, it's not growing, but the cells become smaller. I think really also a very nice time-lapse video. Yeah. So this is It's also quite a nice one, yeah. And the cells become smaller and smaller and smaller, right? And basically, yeah, now the video starts to loop, right? And last week we looked at this one over here, yeah, how it goes on. It continues, and now we can actually see how this will then start, yeah, to develop into into a tadpole, yeah. So you see, time lapse is really something very nice. This is was unthinkable just a couple of yeah, years ago, that hobbyists, amateurs, and so on are also able to do yeah, videos like these, yeah, with equipment that's now readily available. Yeah. So yeah, so that's basically now developing into yeah, a frog larva, a frog tadpole, and it's in just in a second or so it's going to hatch and escape. So very nice video. So just this one I already showed you last week, and so again, very much a big thank you, a big thank you to, yeah, for having sent the video. Okay, and yeah, big thank you to Dave for having made the video available, so that we can share this. But I think I'm just going to show you this again very quickly because it's so fascinating. Yeah, so the 7.3 hours sequence. Yeah, so fertilized egg cell dividing. Now we've got 2 cells. Yeah, and yeah, you see how it's dividing. Yeah, and overall it does not grow in size, but the cells become smaller. Yeah. Really, thank you very much to Dave again, and also to Jesse for those yeah, wonderful videos. Yeah. I think I asked him 2 weeks ago how he did that, and he somehow filmed it vertically. He somehow put those frog eggs somehow between 2 glass plates or something like that. Yeah, but I'm sure there must be several possibilities on how you could do that. Yeah. So yeah, just wanted to share this with you, and I'm going to show you another thing just before I go and answer the questions, because yeah, I've received donations over the last couple of yeah, live streams, and look what I bought. Yeah. This is a slide box, and yeah, it says here basically it's pathological tissues. Basically, it contains tissues with different diseases. However, there's a big however, unfortunately. So there's a little yeah, description here, yeah, of the slides in different languages. Yeah. So, for example, over here it shows here, yeah, different type of even cancers and problems. Yeah, and as a matter of fact, the slides are quite nice. However, there's a huge problem. Not a problem with slides, a problem with myself. And this problem is I know too little about histology to actually make sense of what we're seeing here, right? Just to give you, let's make this a little bit bigger here, just to show you here. Okay. So yeah, it's basically liver cancer, right? But very nicely labeled in different languages. But the problem is, if I put it under the microscope, I'm not able to make sense of it. That's the problem. I'm not a doctor, I'm not a pathologist, so I'm not able to actually explain to you what we're able to see. So I bought this here, was very happy with it, yeah, and but I realized that I have a lot of learning to do, right? And over here, for example, yeah, chronic inflammation of the colon, which is the large intestine. Yeah, but if I show it to you, I'm not able to make a lot of sense of it. I mean, yeah. So what I'm going to do is I'm not going to show them to you under the microscope right now. Okay, there's some kind of a problem of the thyroid gland. Yeah. I'm not going to show it, but rather I'm going just want to tell you that I'm kind of trying to make sense of this and learn a little bit more about those, so that I can actually make some kind of a meaningful video or live stream where I can actually explain to you what we're actually seeing. Yeah, yeah. So this is a little bit a general problem that yeah, with that because I'm not a doctor. Yeah, tuberculosis. Yeah. But what are the things that I'm supposed to actually point out? Yeah, so that's yeah. This one actually is easy. You know what? Why not? I just show this. This is basically when you've got Kohlenstaublunge. This basically means that when people have inhaled some carbon, yeah, then it becomes yeah, the lung becomes black. Yeah, so I'm just going to show this to you. I'm doing this a little bit also as a thank you for the how do you say, for the donations, because when you give me donations, I use it for for buying slides that I can share with you. And let me see if this is, yeah. So what you see over here is, yeah, these are the how do you say, the alveoli, yeah. But I'd have to, I wonder, I don't know, I wonder if these dark patches here is the carbon. But what you have over here, those spaces here, these are the air sacs, yeah, the alveoli for, yeah, of the lung. And over here we've got blood vessels and also the bronchi, yeah, and so on, yeah. But that's basically, these are basically histological slides, and I assume that those black patches that you see here, that this is the carbon, and that's the reason why the person got ill, yeah, by having inhaled this carbon. Yeah, but that's something that I'd have to guess, right? Okay, just wanted to briefly show it to you. Okay, yeah, just wanted to quickly show this to you that this is something that I have also on the horizon that I would like to, once I know a little bit more about histology and pathology, that I would like to share this with you as well. Okay, so what I'm going to do now is I'm going to have a look at some of your questions. Marie just posted something off topic. Your trick with the two polarizer sheets works a treat with mineral slides. The colors are mind-blowing, so thank you. Ah, yes. So this is basically, if you have crossed polarizing sheets, then you can actually do simple polarizing microscopy. Yeah, so basically put polarizer and then a polarizer on the bottom, then you put the slide on top, and then you put another polarizing film over it, and mineral slides look quite nice. So I'm going to quickly, how many pages? This is 1.5 pages. Maybe I already answered some of them. Okay. Interestingly, you call this a pond. We would call it a lake here. Is the technical reason that we call it a pond? Okay, good point. The pond or the lake, actually, there's no specific reason why I'm calling it a pond or a lake. But a little bit of the background here, this pond or lake is actually not a natural one, but was actually made back in the 1930s for gravel. So they basically dug little stones and gravel for construction work. And I did a little research. In 1947, it was actually converted into a swimming lake. Yeah, so it's an artificial pond, an artificial lake. Yeah, because it was used for mining the gravel, little rocks for construction work. Yeah, how the slime mold's doing, at least the one I've got over here, not so nicely, because bacteria are starting to overgrow it. Yeah, are stentors regenerative? We already talked about that. Someone did some research that yes, apparently they are regenerative. I initially had my doubts because it's a single-celled organism. But apparently, they are able to regenerate, and I suppose they are able to do that if maybe the part contains some of the DNA, and those protozoans have multiple nuclei anyway, so maybe that's possible. Yeah, my C sample was complex name, Stentor, not Vorticella. Okay, yes. We feel like keeping Vorticella and Stentor as a micropet is harder compared to Paramecia. Good question. Basically, growing Stentors and Vorticella. What I found out generally is that there is always some form of a progression going on. So this means that one type of microorganism starts to multiply, another one starts to disappear. So there's always some kind of a dynamic change going on, right? So I don't know how easy they are to keep, but generally over long term, I think it's probably going to be unstable. So if you want to keep the microorganisms longer for observation, for example, maybe for over a couple of days, then I definitely recommend that you keep them in this so-called in this moist chamber, something like this. You make a slide, and then you put the slide in here. You probably want to make a rack. I mean, some months ago, a 3D printed one, so that essentially you're able to keep it moist. Yeah, and this way you're able to keep it viable for sometimes quite a surprisingly long time. Yeah, I have not found Stentris yet. Several forms of Vorticella though, and they are just filter feeds. So I see no reason why they wouldn't be easy to keep in the same way that other filter feeders. Yeah, What I've seen is, that obviously they want to have some kind of a place to grow on. So you probably want to provide some kind of a surface, I would say. Yeah, maybe some plants or some decomposing material. Yeah, it's really cool to see the free swimming stentors, though. The ones I've seen online have always been attached to something. Yeah, I mean, because that's when they're attached and when they have this typical trumpet shape, that's a little bit, the way that, they are most commonly shown. Yeah, the size of the stentor, maybe when extended, maybe a millimeter or two. Yeah. A poor man's micrometer slide is to use basically some lead. Yeah. Or for example, there are these leads that you have for these pencils that you, how are they called? Where you push the back part and then a little lead moves out, and they have a standardized thickness of 0.5 millimeters, so you can use this as a reference. Yeah. I just want to share my appreciation of combining technologies to show the different steps. So that's basically this, the reference to the OBS Studio. Yeah. So roughly the size of a copepod and other creatures barely visible to the naked eye. So I think the stentors you should be able to see, but because of the transparency, they might be a little bit difficult to make out. Yeah? No, I'm not familiar with this one. Okay. No, well, if you ask, is it a bit like a euglena? Well, they're all protists. Yeah. So it's really a question of where do you want to draw the line. I mean, they're of course similar because they're single-cell eukaryotes, but they're different. Okay, can you remind us about the stage scales? Yeah, I'll probably make a video about this, how you can use a stage scale, for measuring. They seem to have a similar lifestyle. Are they closely related? Well, the question is, they're protists, they're single-celled. In that sense, they are kind of... You see, in taxonomy, the question is what does closely related mean? It's really a question of where you want to draw the line. Strictly speaking... they're both eukaryotes, and in that sense, they are closely related. And in that sense, even we humans are, let's put it this way, more closely related to them than compared to, for example, we humans are related to bacteria. Yeah, but it's really a question of where you want to draw the line. Yeah, would it be advantageous to use slides with spacer rings? Yeah, I already talked about that. We can try it, obviously, and I would say it depends a little bit also on the on the movement of the stentor. Yeah. Where exactly does the stentor perform phagocytosis? Can it be in any point on the inner surface, or is the specific area? I'm not an expert on this, but they have this mouth area, and I suppose that essentially being single-celled, it's is similar also to the paramecium. They also have a mouth, where essentially they start to form food vacuoles. So in that sense, I would probably say that there must be a specific location where it's actually converted into a food vacuole. Yeah? I so far have done 90% of my observations in 2 milliliter drops with no cover glass. It gives some advantages, but anything fast moving does go in and out of the focus a lot. Okay, yeah. So this is, so basically, sometimes if you want to pick off individual microorganisms with a pipette, then of course you do not want to use a cover glass anyway. Yeah? For what are you using phase contrast yourself? Well, I mean, I usually use nothing specific. What I do usually is that when I observe whatever I see, I simply try the different contrasting techniques, and the one that gives the nicest picture, the nicest contrast, I use for my videos. Yeah, but for bacteria, the phase contrast is of course is very recommended, because bacteria appear to be dark on a brighter background and are much more easily visible. I thought about buying a module for my i4. Don't know exactly. Is i4 is a microscope? I don't know so much about that, about this model. Yeah. Looks like X-ray. Yeah. How long can a stentor live? I don't know. I mean, if you can see this question a little bit philosophically, theoretically, microorganisms live indefinitely, because when time comes, they simply divide and then they're young again. So you could actually say that microorganisms, many of them don't have a built-in lifespan in the way that we humans have. Yeah, yeah, win the lottery to get DIC. Yeah, so basically the leaves were a little bit too thick. We already found that out. Yeah. Flipping them around in some cases can give a nice effect. I also show that with a spicules in them. Okay. Yeah. I thought that Stentor were sessile. Yeah, so they are sessile, means they're attached to surface. They aren't, but as we can see, they also move around. Yeah. I'm quickly going through the questions here. Yeah. Do you see quartz crystal to slow down the rapid retreat? One could reduce the viscosity of the liquid by adding methyl cellulose or proto-slow. As a matter of fact, yes, I agree. And I have over here, as a matter of fact, 2% methyl cellulose solution. So I bought myself online some methyl cellulose powder, added a little bit of water, and indeed, yeah, you can see it's a very thick solution. And when you add this, then it will slow down the microorganisms. Yeah. Yeah, we talked about those. They can detach, and they're able to regenerate. And yes, yeah, only around. I think micromanipulators don't. I hope they don't cost 20,000, but it wouldn't surprise me. Yeah. So vacuoles have several functions. I wonder what drives the creation of vacuoles. There must be some signal pathway. Oh yeah, vacuoles, vesicles. There is the cytoskeleton. So there are these protein fibers that are actually able to change around the shape of the membrane. Yeah, so there is a mechanism. Also, for example, vesicle formation. So there are indeed in the membrane there are proteins that allow for the membranes to form, to deform, and to actually build vacuoles and vesicles. Yeah. So I'm joining a microscopy man says, I'm joining a microphotograph competition. What would you recommend taking a photo of? This is a difficult question. I have the following suggestion. If you're interested in photomicrography, then try to Google the Nikon Small World. It's called Nikon Small World Contest. And they've got essentially beautiful, it's a yearly contest, of pictures. And you will see that, it's not only if the image looks nice, what counts, but if it's also scientifically interesting. Yeah. So have a look there. I would say the following. If you take part of a contest, the point of a contest, of course, is not only winning, but it's also you want to share your pictures. And I would say make pictures of the things that you identify with yourself. Okay? I think that's the whole thing. The point of exploring and using microscopes to explore the environment is because it's also personal growth. I think that's also very important. Take pictures of the things that you are interested in. Okay, I know I'm kind of avoiding the question a little bit, right? But this is a little bit something I would, if you're already asking me, then this is what I would say. Yeah, because the thing is if you participate in a competition, your pictures are going to be probably published in any case. Other people are going to see them, and it doesn't matter if your picture is good or bad, or if it's winning or not winning. You're simply sharing something about the environment and about the world. And I think this learning effect is at least as important. Okay, just an opinion. Yeah. Dave mentions here that frog spawn video was shortlisted in the Nikon Small World in Motion. Oh, very nice. Okay, so Dave Lewis just wrote in the comments that apparently he put it into the how do you say, competition. So there is it's there as well. Eppendorf injection for micromanipulator model costs $22,313 without shipping. Whew. Yeah, I mean honestly, Science equipment is crazy expensive. Yeah. This actually would be, honestly, really? Really? I don't know. This really begs the question: should we not? I don't know. 3D print one. How would you do that? I don't know. Because you see, how? Yeah, this would be an interesting thing. Because the point is the following. Just saying. Getting a tip, how do you call this? This here. Getting this one thinner is actually quite easily possible. What you have to do is you have to take a gas flame and heat up the plastic and then pull it apart. And then you're able to get very, very fine tips. But the question is now, how am I able to move the tip and the pipette around in a very controlled way? Yeah. So that is, and that's what you need a micro manipulator for. Yeah. And these devices are, quite expensive, as we can see now. Yeah. Okay. Jesse. Jesse's basically confirming that what my opinion is you get better shots of things you actually care about and take interest in. It's simply a case of passion. Yeah. Share with the world the things that you're interested in. I would say. And. I have already a couple of ideas on 3D printing one that's going to take me a little while to get through. Honestly, would be great if, but this is probably one of the more complex projects, actually making a micro manipulator. Okay, people, you know what? It's been 1.5 hours, and I'm slowly starting to become tired myself again, I'm going to quickly have maybe a very last look. The slide is already started to dry out, so just a very last look to see if this is the slide, which so maybe I don't even know if we're still able to find it. Yeah, but just a very quick. Yeah, I don't know. This is the slide from before, but it could be that. I don't know where it is. Yeah, but I hope that at least you've seen how these dentures look like and so on, and I think I'm just going to call it a day for today. Where is this again? Yeah, so that was basically, for those of you who joined late, that's basically the video of the Stentor that I made before. And I'm going to call it indeed a day now, because it's pretty late already where I am. I hope that you liked the video again. I do have an Amazon affiliate shop as well, which I think I have to update again a little bit. It's called, you can find it on the microbehunter.store, and then you'll get, if you want to browse some microscopy products. I think I'm just going to leave it at that again. Thank you that you have joined me again this week. Hope to see you again next week. All the best. Happy microbe hunting as always, and bye-bye.